the glycolysis. So in the previous class, we saw the step 1, where the glucose is phosphorylated at the 6th position to obtain glucose 6-phosphate. And because of phosphorylation, one ATP molecule is used, actually is consumed in the first step itself. The second step of the reaction, the second step of the glycolysis, the pathway, is this. Glucose 6-phosphate is isomerized to fructose 6-phosphate. And you know the relationship between glucose and fructose sugars. While glucose is an aldose, fructose is a ketose. However, both of them bear 6 carbon atoms. In fact, they are isomers. Now, the phosphate group's position is not changed. It is still at the 6th phosphate, 6th carbon atom. What is changed? You can see a 6-membered ring is isomerized to a 4-membered ring. I mean, is reduced to a 4-membered ring, but the first carbon atom is a CH2OH. In fact, the open chain structure of fructose, the keto group is on the 2nd carbon atom, is it not? Even the reaction goes through opening of the ring of the glucose into the open chain form, and then swapping of the carbonyl and the alcohol group between 1st and 2nd carbon atom, and then closing into the fructose ring. And the enzyme involved in this reaction is the phosphohexoisomerase. So this enzyme converts one monosaccharide into another monosaccharide. Yes. The third step of the glycolysis is one more phosphorylation. This is the second phosphorylation happening, right? Because the first phosphorylation happened when glucose was converted to glucose 6-phosphate. Now this is fructose 6-phosphate is getting converted to fructose diphosphate, 2 phosphate groups. One more ATP is used up, and the phosphorylation happens now at the 1st position. Here, the 1st position. So the product is fructose 1,6-biphosphate, also called bisphosphate. Sometimes can also be called as biphosphate. Fructose 1,6-bisphosphate. And you know, the phosphorylation happens through the involvement of enzymes called kinases. The enzyme is phosphofructokinase 1. And whenever the kinase comes into picture, the ATP is converted to ADP. Magnesium 2+ is taken as cofactor. Now, this is the step number 4, is it not? And by step 4, even by 4 steps, 2 ATP molecules have been consumed, is it not? And anyways, there is one significance of this step. Why fructose 6-phosphate is converted to fructose 1,6-biphosphate? Because fructose 1,6-biphosphate is specifically targeted for glycolysis. You should know one thing, that all the intermediates and all the compounds of our pathways we are discussing, they can be compounds in other pathways also. See, glycolysis is not the only pathway taking place in a cell at any point of time. There will be many pathways taking place simultaneously and parallelly. And even sometimes these pathways are overlapping. What do we mean by overlapping pathways? The compound in one pathway can also be a compound in other pathway. So fructose 6-phosphate, for example, can enter into other pathways also. However, fructose 1,6-biphosphate is exclusively meant for glycolysis. So this is a very significant step in glycolysis. Why? Because it gives a substrate which continues glycolysis only. Okay. Now, that is one part. The second part is this. Anyways, what is the objective of glycolysis? The glucose has to be oxidized, and in the process, ATPs have to be produced. But a counterproductive thing is happening. A reverse thing is happening here. Why? ATPs are getting used. Do we have now? Glycolysis is meant to give ATPs, but here ATPs are getting used. Why? Why? Because the first 4 reactions of glycolysis is called preparatory phase of glycolysis till now. So what do we mean by preparatory phase of glycolysis? Where actually, the oxidation has not started yet, strictly telling. What has happened, if you look at the structure of glucose and the substrate and the product of fourth step, fructose bisphosphate, look at those structures. Nothing has changed too much. Extra phosphate groups are there added, and the glucose is converted to one more hexose, fructose. That's all. So till now, there is no oxidation at all. In fact, therefore, this is the reason why glycolysis is divided into 2 phases. This is the preparatory phase, which is non-oxidative. It is simply transfer of phosphate groups into substrates as well as isomerization. From now onwards, actual oxidation takes place. Okay, so now you see fructose 6-phosphate, of course, the same thing which I have just repeated here, except same reaction. I'm sorry for this. I have just repeated this. Yes. From here onwards, the oxidation starts. See, glycolysis is a pathway in catabolism, not anabolism. And catabolic pathways are degradative in nature. What do you mean by degradative in nature? Large molecules are broken down into small molecules. So if glycolysis is also degradative in nature, where is that breakage? Your 6-carbon atom glucose should be converted into smaller pieces, should be broken down into bits of smaller number of carbon atoms. The first breakage, and in any catabolic pathway, it is essentially the carbon skeletons breakage that should take place. Not simply functional group interconversions, like alcohol is oxidized to oxygen keto group, or a phosphate group is added, phosphate group is removed. Such minor changes in the structure doesn't account for catabolism. Strictly speaking, catabolism should always involve CC bond breakage. A carbon bond should break, carbon-carbon bond should break. Only then large hydrocarbons or large organic molecules can be cleaved into smaller organic molecules. So in the same line of thinking, even the glucose has to be broken down into smaller pieces. The first CC bond breakage happens now. You see, where is this? Where is the breakage happening? 1 fructose. This is fructose 6-phosphate. 1, 2, 3, 4, 5, 6. Now the carbon-carbon bond between the 3rd and 4th carbon breaks, and then giving us 2 pieces. One is the dihydroxyacetone phosphate, the other is the glyceraldehyde 3-phosphate. Now look at the number of carbons in each residue, 3 in dihydroxyacetone phosphate as well as 3 in glyceraldehyde phosphate. So a 6-carbon atom fructose is broken down into 3 into 2 pieces. And the enzyme which carries out this reaction is aldolase. Anyways, so why is the name aldolase given to this enzyme? Because the reaction of breakage is a reverse aldol condensation. Now you see, I told you, right? Whenever there is a reversible arrow mark in a reaction, the opposite side is also possible. So these 2 smaller sugars can combine and give rise to fructose also back. The same aldolase enzyme can catalyze this. So if these 2 are joining, that will be through aldol condensation. If this is breaking, it is reverse aldol. If they are joining, it is aldol condensation. So since the enzyme can do both, can work either sides in either directions, the name is given as aldolase. In fact, whatever the glycolysis we are studying is only at basic level. We are just getting to know the reactions simply. But every reaction is studied in deep up to the level of its mechanisms. So there is a lot of literature available on how this bond breakage happens. What is the mechanism of the enzyme actions? What are the cofactors involved? So if you are interested, you can study that at least later. Fine. Now you see, till now, what are the things that has happened? A glucose molecule has been broken down into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and 2 ATPs are used. The next step is simply an isomerization step, where you see the dihydroxyacetone phosphate is not going to continue glycolysis. Only the glyceraldehyde 3-phosphate is going to continue the glycolysis. Therefore, all the dihydroxyacetone phosphate produced should be converted to the glyceraldehyde 3-phosphate, which happens in this step, where dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate. It is again simply an isomerization of a ketose and an aldose. The enzyme involved here is triphosphate isomerase. This is analogous to your isomerization, glucose 6-phosphate isomerized to fructose 6-phosphate, right? So you see this, glucose 6-phosphate isomerized to fructose 6-phosphate. In this, an aldose is isomerized to a ketose, 6 carbon, but here a ketose is oxidized to aldose, 3 carbon, of course. The simplest carbohydrate can be formed with only 3 carbon atoms. You require 3 carbon atoms to form simplest carbohydrate. And as an aldose, glyceraldehyde is the simplest aldose possible, whereas dihydroxyacetone is the simplest ketose possible. Fine. So a ketose is isomerized to an aldose in this reaction. So at this point, what is there in our hand? 1 glucose has given us 2 glyceraldehyde 3-phosphate. So henceforth, all our reactions, we are going to show the reaction of only 1 glyceraldehyde 3-phosphate. But actually, that should be, all the things we write should be multiplied by 2, because the other glyceraldehyde 3-phosphate is also having the same reactions as its fate. Is also meeting the same fate, is that same future. Therefore, glyceraldehyde 3-phosphate reactions, whatever we show now, and all the products we get should be multiplied by 2. Yes, that's a very first time actually oxidation is taking place now, where glyceraldehyde 3-phosphate is getting converted into 1,3-diphosphoglycerate. Diphosphoglycerate is also called bisphosphoglycerate. Please follow the change in the structure of the functional groups here, conversions of the functional group. Glyceraldehyde is having aldehyde as its functional group, as well as, of course, there are 2 hydroxy groups. 1 hydroxy group is linked to phosphate. Now, in the bisphosphoglycerate, what is the functional group? I mean, don't worry what has happened. You just look at the structure. This part is same, CHOH, CH2O, phosphate. CHOH, CH2O linked to phosphate. Whereas an aldehyde has become an acid. Although the acid is linked to phosphate, you may not be able to recognize it at the first hand. But if you clear the phosphate here, it is simply an acid group. So essentially what has happened? An aldehyde group has got converted to acid, carboxylic acid. So an aldehyde getting converted to carboxylic acid is oxidation. Is it not? But like in organic reactions, whenever you want to do an oxidation reaction, you invariably require an oxidizing agent. You always require an oxidizing agent. So in laboratory, organic chemistry laboratory, there are n number of synthetic oxidizing agents, starting from simplest like KMnO4 or potassium chromate, even the molecular oxygen itself, or there are many other complicated advanced oxidizing agent like PCC, like lead oxide, selenium oxide. There are numerous oxidizing agents. But in a cell, again, there are numerous oxidation reactions happening here. But which is the oxidizing agents? Whether in a laboratory or inside a cell, oxidation cannot take place without an oxidizing agent. So which is the oxidizing agent? It's a very important question. Now, and understanding here is also very important to you people. So always remember, there are only two oxidizing agents in the cells. While there are a number of oxidizing agents in our organic laboratory, in the cells, there are only two oxidizing agents. And which are those oxidizing agents? NAD plus and FAD. And it is NAD plus which is the oxidizing agents in majority of the reactions compared to FAD. Of course, FAD is also an oxidizing agent. So whenever cell wants to conduct an oxidation, it uses NAD plus. Okay. So the oxidation of aldehyde group in carboxylic acid is taking place. Therefore, an oxidizing agent is required, and that is FAD. And you all know the chemistry of redox reactions. Is it not? Oxidizing agent after the reaction or during the reaction gets reduced. A reducing agent gets oxidized. So NAD is an oxidizing agent. Because of the reaction, it should get reduced to NADH. Substrate is aldehyde, get oxidized to carboxylic acid. So aldehyde to carboxylic acid, NAD to NADH. Okay, fine. So the enzyme which is responsible for the reaction is glyceraldehyde 3-phosphate dehydrogenase. So now, till now we have seen two classes of enzymes. In fact, three. One are the kinases, which transfer the phosphate groups between the substrates. The other are the isomerases, which simply isomerizes different kinds of sugars. And the third is the dehydrogenases. What are these dehydrogenases? They are the enzymes which conduct redox reactions in cells. Redox reactions. Okay, fine. In fact, if you are going to learn enzyme chemistry later, there are about 6 important classes of enzymes, which are classified based on their actions, like isomerases, dehydrogenases, kinases, hydrolases, like that. There are 6 important classes of enzymes, and dehydrogenases belong to one class. Fine. Now, this reaction, what is the output of this reaction? The output is 2. One is 1,3-diphosphoglycerate. The other is NADH. Please remember, NADH. And why the production of NADH here is important, I'll tell you at the end of the glycolysis. But anyways, so at least I will give you a hint here. Please try to link this NADH to your oxidative phosphorylation. Okay, so there is ETC in the cell. If the NADH is oxidized by the ETC, 3 ATPs are produced. So this reaction is having a potential to give you 3 ATPs. Of course, not directly, indirectly, by the oxidation of NADH. Next step. The diphosphoglycerate, you know, is a very high energy molecule. When we study substrate level phosphorylation, we took an example. There are compounds which are much more high energetic than ATP itself. One example was diphosphoglycerate, the other example was phosphoenol pyruvate. So that is the diphosphoglycerate here. This is the diphosphoglycerate. And even I told, how is diphosphoglycerate produced? I did not answer the question there. I'm answering it now. Diphosphoglycerate, which is a very high energy compound, is produced during glycolysis. Fine. So this can transfer its phosphate group to ADP and give you an ATP. Of course, since one phosphate group is lost, the biphosphoglycerate becomes monophosphoglycerate. Of course, you have to specify the phosphate group's position. It is 1, 3 here, and it is 3 here. So that means what? The phosphate of the first in the first position is lost, is given to ADP. And this is an example of substrate level phosphorylation. Fine. You see, ATP is produced directly by the transfer of phosphate group from a high energy compound to ADP. Fine. So for the first time, we are seeing a reaction which is giving ATP in glycolysis. Is it not? So this is one example of substrate level phosphorylation. And because this is again a transfer of phosphate groups, kinases are involved. The enzyme kinase is involved, phosphoglycerate kinase. And 3-phosphoglycerate, which is the product of the previous step, gets isomerized to 2-phosphoglycerate. It is only the shifting of the phosphate group, simple rearrangement from third position to second position. And the enzyme involved in this is phosphoglycerate mutases, a class of isomerases only, but specific to shift of the phosphate groups inside the molecule. Intermolecular phosphate shifts, the mutases are going to take care of it. The enzyme mutases, and Mg2+ is involved. See, whenever there is phosphate group involved in a reaction, always Mg2+ is the cofactor. Whether it is ATP kinases, wherever a phosphate group is involved in the reaction, Mg2+ comes into picture. So the kinases should always be accompanied by Mg2+. Now, one more rearrangement happens. 2-phosphoglycerate is isomerized to phosphoenol pyruvate. What do you mean by this isomerization? It's simply the enol forms and keto forms. Basically, it is tautomerization. So 2-phosphoglycerate is isomerized to phosphoenol pyruvate. Now, if we have, okay, what's the enzyme involved in this? Enolase. You can see this is a dehydration reaction. I'm sorry, it is not isomerization. It is dehydration reaction, because isomerization would have given you another hydroxyl group somewhere. Anyways, this is dehydration reactions. So the hydroxyl group and hydrogen, you can remove these two things as water and form a double bond. Dehydration is an elimination reaction. So all eliminations give double bonds. Additions convert double bond into single bonds. Eliminations convert single bond into double bonds. So the enolases are dehydrating enzymes. So it is converted into phosphoenol pyruvate. What should be the next step? Phosphoenol pyruvate is again a high energy compound which can do substrate level phosphorylation. Look at this, what happens? Phosphoenol pyruvate transfers its phosphate group to ADP, gives us ATP. And of course, kinases are involved. Mg2+ is involved as cofactor. What is that we have got now? Phosphoenol pyruvate is converted to pyruvate because it is lost its phosphate group to ADP, and ATP is got converted to ATP. Now, how many substrate level phosphates we have seen? 2. Which are those? Diphosphoglycerate to glycerate and phosphoenol pyruvate to pyruvate. So at the end of the glycolysis, of course, the formation of pyruvate is the end of glycolysis. If we can summarize the glycolysis, what are the things happening? What is the starting point of glycolysis? Glucose. What are the products of glycolysis now? What are the products of glycolysis? Pyruvate, of course, along with one NADH. Please remember, NADH is something which is produced in the oxidation step. 3-phosphoglycerate to diphosphoglycerate, NADH. One NADH and how many ATPs? This is the question. We have to answer this question. Anyways, let us summarize the glycolysis first. If you look at the glycolysis, this is the preparatory phase where glucose is converted to glucose 1,6-bisphosphate, and this is the payoff phase. What do you mean by payoff? It is yielding results, yielding dividends. That is called payoff phase. And as I told you, after the breakage of fructose 1,6-bisphosphate into two sugars, three carbon atom sugar, all the reactions you multiply by 2. So 2 molecules, 2 molecules, 2 molecules, 2 molecules, 2 molecules. Now let us calculate the ATP yield of this glycolysis. How many ATPs are used in the glycolysis? Yeah, somebody can answer this question. How many ATPs are used in the glycolysis?
2 ATPs.
Good, yeah. Which are those steps in which 2 ATPs are used? The first step, the very first step, glucose to glucose 6-phosphate, and the third step, glucose 6-phosphate to glucose, fructose 6-phosphate to fructose diphosphate. Okay, so I have only 10 minutes more. You can send your names in the chat box. Good. How many ATPs are produced? Tell me.
4.
4. Where are those 2 substrate level phosphorylation steps? Where are those 2 substrate level phosphorylation steps? Here. See here. Diphosphoglycerate to phosphoglycerate. ADP is converted to ATP. But you have to multiply by 2 because glyceraldehyde 3-phosphate 2 molecules are produced at the end of the preparatory phase. And both the molecules are getting further oxidized. Therefore, everything from this step onward should be multiplied by 2. Anyways, so 2 ATPs here as well as 2 ATPs here in the phosphoenol pyruvate to pyruvate substrate level phosphorylation step. Okay, fine. Now, so totally how many ATPs? 2 plus 2, 4 ATPs. What is the net yield of the ATPs now? What do you mean by net? All the ATPs used minus all the ATPs produced. What's the net yield now?
8 ATPs.
No. Why 8 ATPs? See how many ATPs? How many ATPs are produced? 4. How many ATPs are used by the glycolysis? 2. So 4 minus 2, it should be 2. Is it not? Yes. So 2 ATPs is the gain, ATP gain. And we should not forget 2 NADH also. 2 NADH also. Fine. So at this point, we have a rider. We have a condition. What happens to this NADH is the question. Because whenever we account for a pathway, we should account for all its products. Now the glycolysis has given us 2 pyruvate. See, 1 molecule of glucose has given us finally 2 pyruvate, 2 ATPs, 2 NADH. Fine. Now, when we are calculating total ATP yield of glycolysis, we have to consider 2 parameters. What are those? First is whether the cell in which glycolysis is taking place is of aerobic respiration mode or anaerobic respiration mode. Fine. If it is aerobic respiration, which most of the cells are, aerobic respiration, you know, in aerobic respiration, there is mitochondria, there is ETC. So the NAD, what is the net reaction of ETC? What is the net reaction of ETC? Tell me. Electron transport chain. Mitochondrial respiration. What is the net reaction? Tell me what is the net reaction? What is the substrate of ETC?
Oxidation.
Yes, oxidation of what?
Oxidation of NADH.
Good, yeah, very good. So if 1 molecule of NADH is oxidized by ETC, how many ATPs are produced?
3 ATPs.
Very good. So in glycolysis, how many NADH molecules are produced?
2 NADH.
So if 2 NADH are oxidized by ETC in the aerobic cells, how many ATPs?
6 ATP.
Very good. So if the cells are of aerobic mode of respiration, the total yield of ATP from glycolysis is 2 from substrate level phosphorylation. 2 from substrate level phosphorylation. Additionally, 6 from NADH oxidation. So totally 8 in the aerobic mode of respiration. If it is anaerobic mode of respiration, NADH cannot be oxidized by ETC because there is no mitochondria. There is no oxygen, which is the terminal electron acceptor of ETC. So the NADH have to be processed in a different way. Now, that's the one point. What is the advantage of having aerobic respiration? This is the straightforward advantage. What is the advantage? Additional 6 ATPs gain because of oxidation of NADH. There is second advantage. See, whenever we are looking at glycolysis, or whenever we write down a pathway like glycolysis, we should always look at now what are the products formed. We told 2 pyruvates, 2 ATPs, 2 NADH. What are the reactants of glycolysis? What are the reactants? It is not just glucose. NAD plus is also a reactant. It is coming in one of the steps, right? So 2 NAD plus are used by the glycolysis to produce NADH. 2 NADH. If it is aerobic mode of respiration, oxidation of NADH takes place to NAD plus easily through ETC. So NAD plus is regenerated. If all the NAD plus is converted to NADH without regeneration, glycolysis stops. Is it not? Definitely it stops. Why? Because NAD plus is required for the continuation of glycolysis. And if all NAD plus is over, glycolysis stops. Therefore, parallelly, as the glycolysis is happening, there should be some other pathway in the cell which should constantly regenerating NAD plus from NADH. Is it not? In aerobic respiration, there is no problem because there is mitochondria which takes care of this part. Mitochondria has ETC, NADH goes to mitochondria, gets oxidized to NAD plus, comes back to cytoplasm, again continuously used in glycolysis. But in anaerobic mode of respiration, what is that problem comes into picture? What? First of all, there is no ETC. The NADH is not oxidized by ETC. 6 ATPs are not given. So the net yield of ATPs in anaerobic glycolysis is only 2. I should not tell anaerobic glycolysis. The net gain of ATP in glycolysis in anaerobic cells is 2. Okay. As well as there is one more problem now. What is that? How to regenerate NAD+. NADH is there. Because of glycolysis, NADH is produced and its concentration has increased. How to regenerate this NAD+? How to oxidize NADH back to NAD+? This is the question. And this question is a big question in case of anaerobic mode of respiration. Is it not? So in the next class, we are going to address this question. How is NADH regenerated in anaerobic respiration? In aerobic, I told mitochondria does the job. In anaerobic, how is NADH regenerated? Or sorry, how is NAD plus regenerated? Or simply, how is NADH oxidized back to NAD plus for the continuation of glycolysis? Fine. So I'm going to answer this question in the next one.